Thank you for the great introduction. I am Patricia Onyimba, and I'll be presenting on assessment of extractables and leachables data in ANDA submissions. So there will be two parts to this presentation, and I'll cover the first part on extractables and leachables assessment associated with container closure systems, while my colleague, Dr. Patkar, will cover extractables and leachables assessment associated with manufacturing equipment. For this presentation, I will provide examples of some of the most common issues we see during assessment of container closure systems, extractables, and leachables data in ANDAs. I'll also describe the type of information that should be included in ANDA submissions for container closure systems, extractables, and leachable studies, and will then describe how we assess that information in ANDAs. So throughout this presentation, I may refer to extractables and leachables as EL and container closure systems as CCS. So extractables and leachables, as we know, are organic and inorganic chemical entities, and assessment of these chemical entities should be an integral part of your evaluation of the suitability of your container closure systems and manufacturing equipment used for your drug product. As my presentation focuses on extractables and leachables for container closure systems, I refer you to USP 1663 and 1664 for definitions and recommendations on assessment of extractables and leachables. Extractables could come from different sources. Some examples are chemical entities and additives in individual packaging components. Migrants, like ink and label adhesives from secondary and tertiary packaging components and volatiles from cardboard shipping containers, etc. They can also come from chemical substances on the surfaces of drug product manufacturing systems, as well as other sources. Now, USP 1663 provides additional sources of extractables, and it is important that all sources of extractables are considered in your extractables studies. USP 1663 and 1664 do clarify that extractables can leach into the drug product under normal conditions of use and storage, and as a result become leachables. Leachables, on the other hand, are observed in the packaged drug product because of the direct action of the drug product on the source of the leachables. As such, leachables are typically a subset of extractables, and they are mostly derived from the manufacturing and packaging equipment in primary and secondary container closure systems. Migrants have the same definition as leachables for the USP, except that they do cross a physical barrier before accumulating in the packaged drug product. As such, migrants are typically from secondary and tertiary packaging and ancillary components. So both leachables and migrants are of concern due to their potential safety risks to patients and potential compatibility risks for the drug product. Therefore, they should be evaluated and controlled in the drug product whenever necessary. And in order to do this, it is necessary to know their identities and the levels to which they will accumulate in the finished drug product over shelf life. So chemistry, manufacturing, and controls information in ANDA submissions are reviewed within the Office of Pharmaceutical Quality, OPQ. Now the two offices shown here, Office of Life Cycle Drug Product, OLDP, and Office of Product Manufacturing and Assessment, OPMA, are responsible for reviewing EL studies within OPQ. And while OLDP reviewers are responsible for assessment of CCS extractables and leachables information, OPMA reviewers are responsible for assessment of manufacturing equipment, extractables, and leachables information. So what are the common issues we see during assessment of EL studies information in ANDAs? The information on this slide is not all inclusive, but we do see ANDAs that should clearly include extractables and leachables information for the drug product. However, the information is not included. Sometimes extractables data is submitted, but leachables data is not submitted, or inadequate leachables data is submitted. We also see the use of incorrect safety concern threshold, SCT, and incorrect analytical evaluation threshold, AET. We see where extractables and leachables data is submitted, but there is no information on the method and method validation, or inadequate method and method validation information is submitted. And lastly, for some ANDAs, leachables data may be submitted with impurity levels that are higher than AET. However, no justification is provided for the high levels of leachables, or inadequate justification is provided. Now, these gaps result in a lot of back and forth between the agency and ANDA holders during ANDA assessment. And my hope is that you utilize the information presented today to minimize these gaps. So what do we expect for your CCS extractables and leachable studies? For your extractable study design, we'd like to see that you followed USP 1663 recommendations, that the study is designed to identify all potential leachables, including inorganic, volatile, semi-volatile, and non-volatile organic compounds. We encourage you to obtain information from your CCS suppliers or manufacturers to help you identify which compounds from the CCS components are most likely to leach into the final drug product. Also, your extraction solvents should be selected based on USP 1663. And of course, other solvents may be used with justification based on the drug product formulation and composition of the CCS. In addition, the critical study variables such as extraction time, temperature, volume of the extracting solvent, etc. should be appropriately justified. Your analytical methods should be suitable for their intended use with appropriate detection and quantitation limits. And given the diversity in the chemical nature of extractables, multiple methods may be necessary to identify and quantify all potential leachables. And lastly, all extractable impurities above AET should be structurally identified per USP 1663 and quantified, and the AET should be calculated using the correct MDD and SCT. Similarly, your leachable study design should follow USP 1664 recommendations. The study conditions should be adequately justified. Ideally, you should conduct your leachable study using actual drug product and actual packaging and delivery systems, and any use of simulation solvents instead of drug product should be adequately justified. Your analytical methods should be fully validated with appropriate LOQ, and ideally the methods should be same or similar as those used for extractable studies. Also, you should structurally identify and quantify all leachable impurities observed above the AET, and your AET should be calculated based on the correct MDD and SCT for the product. And finally, the study should be performed using finished product from 3 exhibit batches, monitored through shelf life at multiple time points during long-term and accelerated stability studies, unless adequate justification has been provided. Now that we've talked about what we expect to see for your EL study design, let's talk about what you should include in your extractables study report. Your report should include the initial risk assessment for your approach. It should also include the data generated from your extractable studies, which consider both the primary, secondary, and tertiary packaging components. It should include information on your extraction conditions. Additionally, information on analytical methods and verification of at least the LOD and LOQ should be included. And finally, you should include assessment of the resultant extractables profile. Similarly, your Leachables study report should include data from your 3 primary stability batches, each of which should be followed through expiry, as leachables can accumulate over the entire shelf life of a drug product. The report should include information on analytical methods and method validation studies. It should also include safety assessment of any leachables that are above the AET or SAT. Finally, acceptance criteria should be proposed in the drug product specification if leachables are observed above the AET. Well, now that you know what should be included in your extractables and leachables report, I'll talk about how we evaluate the EL data in your UNDAs. First, we use a risk-based approach to determine if controlled EL studies are necessary for a particular drug product and the level of scrutiny that should be applied to the information submitted in the UNDA. This slide shows Table 1 from the FDA guidance on container closure systems for packaging of human drugs and biologics. And as can be seen from the table, the degree of concern associated with the route of administration for a particular product could be classified as low. However, the likelihood of packaging component dosage form interaction for the same product could be classified as high. And my example here uses something like topical solutions and suspensions or oral solutions and suspensions. So if we determine that EL study is necessary for a given product, we will request that this information be submitted in the UNDA if it is not already submitted. We'll evaluate adequacy of the extractable study design and analytical methods used for the study. For example, we check to see if the study conditions like solvents, temperature, time, duration, stoichiometry, etc., analytical methods and method sensitivity are adequate, and if the study was performed per USP 1663. For method sensitivity, are the LOD and LOQ reasonable, and was AET calculated based on correct MDD and SAT? Do methods adequately cover inorganic and organic impurities, including volatiles, semi-volatiles, and non-volatile compounds? Are we confident in the values reported? We also check to see if any extractable compounds are observed at a level greater than the AET, and if these extractables are adequately identified and quantified, and if leachables data is submitted. If leachables data is submitted, are the study conditions, AET, methods and method validation, number of batches, etc., acceptable? Are leachable impurities observed above the AET? And if so, are they structurally identified and quantified? And is adequate qualification data submitted to justify the observed levels? If qualification data is submitted, we typically consult the information to our PharmTox group for evaluation. And if they find the data unacceptable, then the CCS is deemed not qualified. So now let's talk about CCS changes, pre or post approval. If you make changes to your CCS during under review or after product approval, we expect that you will use a risk-based approach for your EL studies, and your approach should consider things like product risk. For example, is there a change in formulation, manufacturing system or process, etc., that could change the leachables profile of the drug product? Does your CCS change involve a new CCS that was not previously qualified? If the answer is yes, then the EL risk is high and full EL studies will be needed. Or, does your CCS change involve a CCS that was previously qualified? If the answer is yes, and if the extractables profile of your changed CCS is similar to that of previously qualified CCS, then the risk is low and leachable study may not be needed. However, if the extractables profile of your changed CCS is not similar to that of previously qualified CCS, then the risk is high and leachable study may be needed. So I have two challenge questions for you. And the first question says, from ABC is developing a topical cream drug product that contains about 78% purified water and 20% organic solvents in the drug product formulation. Are extractables leachable studies for USP 1663 and 1664 necessary for this product? I will give you a few seconds to consider which answer you think is correct. So if you chose option C, that will be the correct answer. The second question says, from XYZ has developed methods for extractable studies of the container closure systems for packaging of their proposed ophthalmic suspension drug product, but did not validate the methods prior to the study. Should firms' methods be validated? Again, I'll give you a few seconds to consider which answer you think is correct. Now, if you choose option B, that will be the correct answer. So congratulations. We've all been paying attention so far. This slide contains some references on the topic for your information. To summarize what we've learned today, first, a risk-based approach should be used for EL studies of your CCS. Secondly, your EL studies should be performed for USP 1663 and 1664. Thirdly, you should evaluate your CCS extractables and leachables from a patient safety perspective to ensure safe exposure to leachables in the drug product. Lastly, the EL studies information in your ANDA should include initial risk assessment, EL testing data and reports, and toxicological risk assessment of any leachables above AET. And of course, AET should be based on the correct MDD and SCT. In closing, I encourage you to ensure that your extractables and leachable studies are adequately designed and executed per USP 1663 and 1664, and that adequate information is submitted in your ANDA to justify safe levels of leachables in your drug product. I would like to thank the following individuals for their continued support. Binkai, my division director and my boss, Pahala Simamora, the director of DLBP2, Andre Rohr, the senior science and policy advisor for OLDP, and Susan Rosenkrantz, the director of OLDP, and of course the SBIA organizing committee. That brings me to the end of my presentation, and I thank you for your time. I'll be back a little later to answer your questions on the topic. Please submit your questions in the pod for later. I will now hand it off to Dr. Patkar to talk about assessment of extractables and leachables associated with manufacturing equipment.
Good afternoon, everyone. My name is Kshitij Patkar. In this part of this talk, I will cover assessment of manufacturing process extractable leachables data in ANDA submissions. Manufacturing process extractable leachables are similar to container closure extractable leachables, but they originate from process equipment, polymeric surfaces, such as filters and tubing. What are we covering today? So I'll talk about the significance of the assessment of manufacturing process leachables. I will talk about what are the most common deficiencies that we see in ANDAs, what information we assess, such as protocols, procedures, data, mitigation strategies. How do we assess? How do we determine that the provided information is adequate? And in the end, I will summarize my talk. So let's talk about the importance of manufacturing process leachables. Leachable impurities can have significant impact on safety and quality of the drug product. They have the capacity to leach into the formulation and be delivered to the patient. Therefore, their risk should be determined. Also, it is a GMP requirement that manufacturing equipment should be compatible with the drug product, as well as it's safe to carry out the manufacturing process. There are a couple of regulations listed here, 21 CFR 21165 and ICHQ 7511. I recommend that you take a look at these for their recommendations. Now I'll talk about the common deficiencies in ANDAs regarding extractable leachable data. So one of the deficiencies that we observe is that not all relevant contact surfaces are included in the study. Sometimes only the filter extractable studies are provided, but there is no data on the tubing. Sometimes we see there is little correlation between extraction conditions used in the studies and the actual manufacturing process. And I will talk about this in a little details later. We often see only qualitative data, but we don't see any quantitative data. Or we don't see any toxicological justification or data or leachable studies for extractables that are above analytical evaluation threshold. We often see analytical procedure information is inadequate. There is not enough details provided. And we also see applicant sometimes provides a risk assessment with final conclusion, but there is no data to support those conclusions. Now let's see what information we assess in ANDA for these studies. For extractable studies, we assess study protocol. These include relevant polymeric surfaces, solvent selection, extraction conditions, whether they are representing the worst case manufacturing process conditions. We assess if extractables are identified and if they're quantitated. We also assess what mitigation strategies are provided for extractables that are above AET. That could include toxicological justification or leachable studies. If the applicant provides leachable studies, then we see if the leachable studies based on the extractable studies. And that means if applicant evaluated all extractables that were found above the analytical evaluation threshold for their presence in the final drug product. We assess analytical procedures for their details, sampling. We also look at any validation reports that may be provided or the list of reference standards that are included. And then we also assess if there are any mitigation of leachables found above AET is provided. So let's talk about agency's expectations about the data that is provided in the submissions. We look at three things. We look at adequacy of the study protocols. We expect that the applicant provides a risk assessment of the extractables that are above analytical evaluation threshold for their potential risk as leachables in the final drug product. We expect the applicant to use appropriate safety concern threshold. As Patricia already discussed in this presentation, a correct MDD and SCT should be used to calculate the AET. We expect the applicants to provide mitigation strategies again for extractable leachables above AET, as I said before. Now, how do you ensure that the study protocol is adequate? Well, make sure that all polymeric surfaces that are in direct contact with the bulk are covered in the studies. That includes any filters, tubing, and also if there are any polymeric bags that hold the intermediates should be included. It is very important that all of these equipment are included because we often find in ANDAs that a lot of times tubing or polymeric bags data is missing, and then that results in multiple rounds of information requests. Use of appropriate exposure or surface area is also very important. For example, tubing extractable data provided for only a small section of tubing used in the extraction. but the data is not extrapolated to the total tubing that is used in the process stream. Use of appropriate incubation condition is another important issue that we often see in ANDAs. It is expected that applicant uses exaggerated conditions covering worst-case manufacturing conditions of duration, temperature, and solvent. So, for example, extraction using hexane at 90 degrees is really not appropriate for a filter extractable study when the formulation contains only 5% ethanol and the filtration is actually carried out at 25 degrees. Applicants should make sure that extraction conditions are relevant to the proposed process and formulation. For leachable studies, on the other hand, applicants should use drug product or a bulk solution and the conditions that are representative of the proposed process. So for example, if total filtration time is 24 hours in the proposed process, it is expected that incubation time should be at least equal to or more than 24 hours. The selection between bulk solution and the finished drug product for these studies depends on what extractable compounds or the source that you want to study. If you're investigating the compounds potentially coming from tubing, from filling operation, then it's appropriate to use the final drug product. Whereas if you want to look at the filtered leachables, then one can use a filtered bulk solution for the studies. As we said before, the use of correct AET based on appropriate MDD and SCT should be used in the risk assessment. We expect a quantitative data be provided for extractables, especially those are above AET. And once you find the extractables above AET, we expect that applicant evaluates their potential to leach in the final drug product, either using appropriate leachable study or at least provide a pharmacological justification. So once you find extractables above AET, we expect applicant to provide leachable study. Again, the leachable study should be adequate or a pharmacological justification with the data to show that extractables that are above AET do not pose risk to the patient. Once you carry out leachable studies and you see there are leachables above analytical evaluation threshold, then applicant has a few choices. Either provide appropriate process controls with supporting data to reduce leachables below safety concern levels. Some of the examples could be using alternate filters and tubing that actually has less potential to leach compounds into the drug product. Or there could be a pharmacological justification with data to show that the leachables above AET do not pose risk to the patient. Another approach that could be employed is to control the leachable impurities at a certain limit throughout the life cycle of the drug product. But of course, for that, applicant should provide the data and justify the limits at which these impurities are controlled. In summary, I would like to reiterate that the risk for manufacturing process leachables should be assessed based on the formulation and the process conditions. These studies should be performed on all equipment that are in direct contact with the bulk formulation. Extractables above AET should be justified for their levels, either by tox or leachable study should be provided to demonstrate that these extractables are not present in the final drug product above safety levels, concern levels. Any leachables above safety concern levels should be assessed for their toxicological risk and adequate mitigation strategies. So I have a couple of challenge questions for you to see what we have learned today. So question number 1 is, the drug product formulation contains 20% ethanol at pH 6.5, and the sterile filtration is carried out at 2 to 8 degrees. The extractable study was carried out using 1 normal HCl and pH 2. Which of the following statement is true? A, the extractable study represents the worst case scenario for the process leachables. B, the extractable study does not represent the worst case scenario for the process leachables. I will give you a moment to think about these options. Okay, if you choose option B, then you're correct. The extractable study really does not represent the worst case scenario. That is because if you look at it, the formulation actually contains 20% organic solvent at a neutral pH, but the extraction conditions uses strong acid and it's an aqueous condition. So this solvent is really not going to give you correct information about the extractable leachables. Question number 2. The tubing extractable study data shows total non-volatile residues above AET. Which of the following steps are considered appropriate toward mitigation of risk of potential tubing leachables in the final drug product? A. Identification and quantitation of individual compounds from the total NVR. B. A leachable study that evaluated a few known tubing extractables in the final drug product. C. A leachable study that evaluated identified compounds from the extractable studies in the final drug product. D. Options both A and B are included. Well, the correct response is D, because we want to see identification and quantitation of individual compounds, and then we want to see if these identified compounds above AET are evaluated in the leachable study for their presence in the final drug product. That concludes my talk. I would like to acknowledge my branch chief, UI Lee, my coworker and branch chief, David Anderson, division director, Ying Zhang, and SBIA organizing committee. And finally, I would like to thank you for listening to my talk. I will